around the best person that we could lay our hands on in that area was none other than Professor Momina from the University of Nepal. Thank you. The student will be able to be updated on the latest research in cancer diagnosis, and that will help them as they build their careers in medicine, in pharmacy, in nursing, and other careers in healthcare. Thank you.
I'm Professor Momia Condorelli from University of Naples, Federico II. I came here for the first time in this university to promote international collaborations, and also I'm the coordinator of a master's degree in medical biotechnology. So I come here to select some students that will have the chance to come to Federico II and study with us. Also, I hope that this will be only the first of different visits from myself and other colleagues from Federico II to promote exchanges between our university and Kenyatta universities.
Do you think this research that you are doing should be implemented by policymakers in different governments?
I think the research is very important when is sharing within different countries and also with different expertise. So I'm a medical doctor, I bring my expertise, and I also need some other expertise from other colleagues and national and international colleagues. So I really think that this collaboration brings us new results for cancer research.
What are some of the achievements that you have noted?
I am trying to set up a specific biosensor that able to be applied for early cancer diagnosis. In particular, I started with breast cancer, but I can apply this to different kind of cancer.
My name is Mothoni Faith, and I'm going to be your master of ceremony for the day. I hope we are going to have such an interactive session. It's an interesting time that you're going to have together. And before we even start, I would like to ask you, how many people know anyone who has been diagnosed with cancer? Hands up. Okay, yeah, thank you. How many people have lost their loved ones because they had late diagnosis of cancer? Thank you. So I really hope that by now you realize that this lecture is not just any other lecture, but this lecture is something that is for you to know how we can have early diagnosis of cancers through Aptamer. I know it sounds like a buzzword, right? Aptamer, biosensors and diagnosis of all this so that we can detect cancer earlier on. In basic terms, the Aptamer is a missile. So this missile is focused on its target. So it has laser focus on its target. And this target could be an exosome, it could be whatever it is. It could be a cancer cell, whatever it is. And the Aptamer is made up of either an RNA or a DNA strand. I think we are familiar with RNA, DNA strands. Yes, thank you. Even as we study this is not fiction. These are things that are being done and have been done, and we are pleased that we have people who are coming to tell us more about that. So at this juncture, I would like to acknowledge all of you. Let me do a roll call. Medical students in the house. I start with pharmacy. Okay. We will give them the honors. Pharmacy student in the house. Raise your... Okay, clap for them. Thank you, thank you. Medical students in the house. Oh, clap for them as well. Nursing students in the house. Oh, clap for them. Medical labs in the house. Oh, nice, interesting. medical biotechnology in the house? Oh, nice. Have I forgotten any other? Food and nutrition, excellent. I think they are watching us online. Yes, thank you. Let's appreciate them. Maybe Jody will tell us how many people have joined us online. I truly appreciate that you honored this invitation, and I know many people are coming. So that we can have this timely conversation. Now, I would like to take these honors to also acknowledge our faculty. Faculties in the house. Oh, let's appreciate them. Oh yeah, my lecturer, Dr. Kizito. Thank you. And now, I would like to take this time to introduce someone, an individual who has done a great work in inspiring innovation and research and biotechnology. And he's someone who's very passionate in both mentoring people, but also in his work in evidence-based medicine. Maybe you might not know all of that, but you know him as someone who has offered his shoulders for you to step up and see the world of research. So this is none other but Dr. Kahiga. Put your hands together for him. He's a chairperson of the Department of Pharmacology and Clinical Pharmacy. Put your hands together.
Thank you, Mudoni, for the kind words, and just to acknowledge other people outside the university who are in the audience. Just by a show of hand, anybody outside Kenyatta University? Consultants, practicing people outside? None. Okay. Very pleased to be here and to host this very important discussion. And I know we are all looking forward to hearing where we are with cancer. And as the MC has said, I know all of us have been affected in one way or another. And we are very, very delighted to have with us one of the greatest scientists in this area of cancer, all the way from Italy. And allow me to just do a brief citation of our guest. Our guest name is Professor Gerolama Condoreri. She tells me she has a shorter version of that called Professor Momina from the Department of Molecular Medicine and Medical Biotechnology from Federico II University in Naples. And Professor Momina is a full professor of general pathology, and she has worked in various places. About nearly 30 years ago, she was working with an institute called CNR Research in Nepal. I guess it, from the word I see, it deals with endocrinology. She did her PhD in cellular and molecular biology and pathology, and in 1997 officially certified for endocrinology and metabolic diseases at University of Nepal. Ladies and gentlemen, let's put our hands together and welcome Professor Momina to give her talk.
I'm very pleased to be here in this wonderful country. Today I'm presenting, I would like just briefly to present something to my university. I come from University of Naples that is called Federico II. Our university was founded 800 years ago by this emperor called Federico II. My lab So, Department Medical Molecular Medicine, Medical Biotechnology. My lab is involved in studies in molecular cancer, especially where we're looking at role of microRNA in cancer, role of exosomes in the crosstalk between cancer cells and microenvironment. We also look at role of cancer stem cells in the behavior of cancer and the response to therapy. And this is the topic of the talk. So since a few years we work on this field that is called Aptamers. So Aptamers come from a library. So they are artificial molecules. They are not molecules present in biology, but it's a library that we have. Once you recognize and select your molecules, you can order. So you can have biotechs that can synthesize your aptamers. We can see that aptamers versus antibodies, the difference, the advantages is the cost, production and flexibility of nucleic acid comparing to antibodies. How can we identify our sequence? So this is a process that we do in the lab. So the first step is called SELEX. Means systematic evolution of ligands of exponential reach. This is a very variable sequence, but has two fixed region that is useful for amplification. For PCR, so 5' and 3' recognition of primers. In our lab, we raise aptamers for different purposes: breast cancer, glioblastoma, rectal cancer, pancreatic cancer, lung cancer. Some are in a more advanced stage of research. So there is another opportunity that we are exploring that is called liquid biopsy. I don't know if you ever heard about liquid biopsy. Do you know something about in the audience? So usually we do biopsy in the tissue, but very recently you can think about or recognize characteristics of the patients taking blood. It's much easier to take blood of a patient instead of doing nodule biopsy. So that's why it's called liquid biopsy. We are also studying in my lab the role of extracellular vesicle in crosstalk between cancer cells and microenvironment, for example, fibroblasts. So we are studying the way how the exosome from breast cancer are released, are uptaken by fibroblasts that become aggressive. They can change the phenotype, cancer-associated fibroblasts, and increase the aggressivity of the tumors. We want to know by our process if there are some proteins specific in extracellular vesicle from breast cancer. So we apply the SELEX. Okay, so the SELEX means that we isolated the breast cancer from the tissues from human undergoing to surgery. We are able to apply primary culture, and the cells are growing, and we take medium of the cells that contain extracellular vesicle. In this case, medium is like blood, okay, it's a liquid. And as I told you before in our flow chart, so this is the SELEX and this is the second step, that is the analysis of the sequencing coming from the deep sequencing, bioinformatic tools that build up this sort of tree. Seems scaring, these figures, but reality, I mean, each line is a different aptamer.
Access to knowledge, solution to unanswered questions.
Guidelines to pursuing your rights and repossessing your property, intellectual or material.
This and more only on Law Matters.
We need to increase specificity and to set up an assay that is more reproducible, that say yes or not, cancer or not cancer. So this is a trend that is encouraging, but we were not satisfied by this assay. So we start a collaboration with the physics department of Federico II, colleagues that actually will come in Kenya next week as well, to design an Aptamer-based biosensor. So I am learning about biosensor because I'm not a physicist, so was identified by this is a pioneer of biosensor, father of biosensor, Professor Leland Clark, try to convert biological signal into a measurable signal. So a good biosensor should provide accurate, reproducible results, be cheap, small, portable, and the reaction should be independent by other physical parameters. So there are part of biosensor, sample analyte, what we want to test. Then bioreceptors that are recognition molecules that can be, for example, aptamers, can be enzyme, antibody, cell, and signal transducer and electric signal. So sensor that you know, for example, pregnancy test is a biosensor, or glucometer is a biosensor. So we won't try to arrive to this very simple test. We are not close, I mean, we are not at that point, but I hope that maybe in a few years we can go close to this point for also breast cancer liquid biopsy with our aptamers. So biosensor can be applied for many reasons, environment, medicine, drug discovery, etc. And Apta sensor use aptamers as recognition molecules. So we are applying colorimetric Apta sensor. In which way? So my colleagues used these gold nanoparticles that have the properties change the colors when they are linked to the molecules. So what we did in collaboration with physical department is to link our aptamers that we identify from the breast related extracellular vesicle with the gold nanoparticle. So the first step is link gold nanoparticle to aptamers. This is possible because we order the aptamers with a special link that is able to bind gold nanoparticle. Okay, this is something that we do in collaboration with the people with physical department, and they are able to see, to follow by optical density if the binding of the aptamers with gold nanoparticle was successful. They are happy only with only this very little red shift. That means that gold nanoparticles are linked to the aptamers. So in our solution, the first step is to induce aggregation of gold nanoparticle with aptamers. And we have the development of a signal, optometric signal, means that the solution develop this blue signal, blue color, okay? Then what's happened? We add serum from breast cancer patients containing extracellular vesicle. In this case, extracellular vesicle displays the binding of the aptamers to the nanoparticles, okay? Extracellular vesicle reduce the binding and the signal from blue become red, okay? So you have a decrease of signal, and you can detect this signal, this color, okay? And this is a very simple methodology. So here in the red there is the control, and in all the blue whale is the breast cancer. So in this case I can show the we analyze serum from 100 samples, 50 tumoral and 50 non-tumoral samples. You can see that tumoral has a lower color development comparing to the normal, and this assay is becoming more specific. So if we calculate the specificity is 98%, we have only one non-tumoral that are in the same of tumoral, and we have only few samples that are false negative, okay? So we have to work on this increasing sensitivity, but I think we are in a good route, and also we are trying to test other tumors, for example lung cancer, with the same methodology. Do not cross-react so with breast cancer. So this assay is very similar, very specific for breast cancer. The next step is to develop, also my colleagues wants to use in artificial intelligence to calculate so we are calculating the area under the curve, and so they want to calculate with artificial intelligence and this and to render this more exportable to all the labs and also in the clinic. I don't know if you have question for this part because I can have another example of Aptamer application. If you have some question regarding this part of breast cancer. I don't know if I was clear or I tried my best, I don't know, but I can take all the questions.
Thank you, Marilla.
If you are tired.
Any questions?
also
Application? I'll go first. Okay. So in this regard, you've tested on lung cancer and breast cancer. Ultimately, do you think this would work with any type of cancer? I'm very interested in pancreatic cancer that normally it's delayed, right?
Yeah.
Normally found at stage 4. So would this also target?
Yeah, actually we are trying to do with other models like pancreatic cancer and also there is a very important needs of primary screening, so pancreatic cancer, lung cancer, and rectal cancer. So we are doing, taking advantage of our experience. We are doing this approach for other models. So we have each PhD has his own project. Each PhD has a project with several models. We are lucky that we collaborate also with clinicians that give us samples. So this is another important collaboration with clinicians that, for example, breast cancer, we need many samples because breast cancer is heterogeneous, has very different subtypes. We want to know if our methodology is more suitable for one subtype or for another one. And also we want to apply this for follow-up of therapy, not only at time 0, but if a patient is positive at the beginning of his disease and starting chemotherapy, we want to follow the patients during chemotherapy and see if they become negative, and also follow for, I don't know, few years, and to discover if a patient become positive only with the blood test can be useful to avoid to not recognizing recurrence of the tumors. So we want to apply also for follow-up of therapy.
Okay, as she's going to take that microphone around, I would like to ask, does this have a genetic component to it, such that is it applicable to a different genetic race?
I mean, we don't know. We really need many samples to go into details of genetics. For example, in breast cancer, we can see if the mutation of BRCA1, BRCA2, we can look at this, but they are not less common than the other. So we don't have many samples. So it's important. Is there this having samples? Another collaboration we can do with the universities to have clinicians that take sample for us, and together we can do the studies in measuring and see if the positivity or not.
All right. Okay, there's a question there.
Yes.
Okay, I will take this in.
Thank you for the presentation so far. My concern is on the stability of the target and the aptar.
Yes.
Being a ligand, the complex that is formed is quite stable. How is the reaction reversed?
Yeah, I mean, the aptamers, we study stability. The way we do is we take the aptamer, incubate with serum of several hours, and we see, then we run in the gel, and we see if there's degradation. And we saw that our aptamer is pretty stable. The binding to the target is also stable. We can, and also rapid. We can just incubate 15 minutes, and we have the recognition and the binding of the aptamers and the target.
Hi, thank you so much for the presentation. I have two questions. The first one was when you were explaining how the colorimetric apta sensor works. Step 2, when you said you add serum from a breast cancer patient to the extracellular vesicles that have now bound to the apta, what is making the color change? Like, how are we getting to the color change?
Yeah, it is something that I'm learning as well, but essentially the color change is done because when aptamers aggregates with gold nanoparticle as this, so it's a gold nanoparticle that has developed this color. That is with adding a particular reagents, chemical reagents, develop the color. So it's particular reagents added to the solution develop the color with the gold nanoparticles. Instead, when the aptamers, when the exosomes recognize the aptamers and are added, they displace, so they compete. Okay, so they compete, the exosome compete with aptamers, with gold nanoparticles, and displace the binding, and so the colors reduce.
Thank you. And then the second question is, can the apta sensor stage the cancer, because that's really important in determining.
Stage.
Yeah.